Well, it's terrific to be able to join you and present on spatial temporal omics, multi-omics, in 3 and 4D imaging. And I'm going to make the argument throughout this short talk that we need on the order of 20 to 50 nanometer resolution for a variety of reasons for this project. What are those reasons? We need more barcode reads per square micron. We need, this will be roughly 1,000 times more in 3 dimensions. We need to be able to study epigenetic marks at the DNA and protein level, keeping in mind that the nucleosome itself is 20 nanometer in diameter. For brain connectome, synapses, dendritic spines, disambiguation of adjacent synapses is 50 to 150 nanometers. We think this dense data configuration results in lower cost, including and partly mediated by lower error rate. How do we do this? We want to use thick sections, not compressed monolayers or averages over thick sections. We want actual thick sections within disease stacks. So 20 to 50 nanometers over 20 to 50 microns. How do we do it? Molecularly, oligo paints, technology pioneered by the Wu lab at Harvard, Ting Wu's lab, DNA exchange imaging from Peng Yin's group, and or expansion microscopy from the Boyden group. We use combinatorial fluorescent identifications. This doesn't have to be full de novo sequencing, but sequencing by hybridization, ligation, and synthesis by polymerase. We've seen this exponential curve for 1D sequencing that's gone over 20, maybe 60 million fold in just a decade or so, and going in improved quality and clinical relevance, going from haploid genomes, which were clinically irrelevant, to diploid genomes, and $300 per genome has been in place now for 2 or 3 years. Rare genetic diseases are handled by whole genome sequencing for the most part. Common diseases provide us a way of spreading out the R&D cost, bringing the cost per, say, gene therapy vaccine down to $2 per dose. Another example of this, which is one of the themes of the meeting, is age-related diseases. These affect everybody on the planet that manages to escape early accidents and infections. And these include 4 papers that we published on, 3 on AAV and 1 on CMV. Noah Davidson has used this to launch a company called Rejuvenate Bio, aiming on these combination gene therapies for multiple age-related diseases and multiple mechanisms and pathways for aging. Now, we'll maintain that essentially all fluorescent next-generation sequencing is microscopy. It is not something that you can use to streamline microscopy. It includes microscopy, and that goes back to our first paper in 2005, where we used a Nikon microscope to do the sequencing by synthesis and sequencing by ligation. The G007 in 2007 and modern NovaSeq and MGI, cool MPS technologies. These are all imaging, but in monolayers, so nanometer thickness. We need much thicker specimens, so we get thousands of times more reads per square millimeter. So fast forward a little bit further, and we have 2 major instruments that my lab is familiar with. One is the read core, recently acquired by 10x Genomics, and the Brooker Vertara, co-developed by Ting Wu's lab at Harvard Medical School. This is useful for DNA, RNA, been proven for DNA, RNA, and protein at some of them at super resolution, as high as 0.2 nanometer in very special DNA origami cases, but 20 nanometers is fairly routine for actual in situ tissues. ideally removing fluorescent background via polymer embedding and clearing. So I'm going to show you DNA, RNA, and protein in quick succession. This is an example of two small pieces of blow-ups of a piece of chromosome 19 showing the maternal and paternal chromosomes and the color coding of the walking along the chromosome, which has now been done by the Wu lab for all of the chromosomes simultaneously, and increasingly higher and higher base pair resolution. So we should get used to whole genome imaging, not just of the folding, but of the associated proteins, as we'll see in a moment. This is an example of an even closer blow up of a single loop, a short 270 kilobase loop, closed by the orange and red 10 kb loop closure. And each of these dots is a single molecule of fluorescence imaged using oligo paints. The cartoon at the top showing a hybridization with a black region and two red and yellow fluorescent exchange regions. Again, so it's DNA, this is RNA. Each dot is a single RNA molecule changing color at GATC, but not position. And then you can have the computer recite what it saw through the cycles of fluorescent sequencing. And this is these are from Personal Genome Project #1. That's my fibroblast. That was a single fibroblast cell from Jay Lee and Evan Doherty. And then finally, proteins, DNA, RNA proteins. This is done by DNA exchange imaging, where the protein, the antibody, is labeled with a short DNA, maybe 9 nucleotides long, and then it is imaged by transiently binding fluorescently labeled short DNA. This can be a full Y-shaped IgG antibody, or it can be a little domain of the single domain, which is much smaller and hence higher resolution. The diffraction limited on the left, super resolution DEI on the right. You can see that you cannot resolve the synapses in this brain specimen without the super resolution. You need on the order of 20 to 50 nanometers in order to see which, when you have a lot of close axonal and dendritic spines, that to tell which is which, you need to identify the synapse, the pre and post synaptic proteins here. We can do multiple proteins at once. We don't know what the upper limit for the number of proteins, but it's probably similar to DNA and RNA, tens of thousands per cell. And you can also tell whether it's excitatory, inhibitory, which is important functional information in this case. This was done by Yu Wang, who was a graduate student and postdoctoral fellow in my laboratory, and in Peng Yin's laboratory. Finally, we want to do time axis, and doing it real time in living tissue like this zebrafish larvae is highly limiting. It's limiting both in time and in space. It's hard to do more than a fraction of a cubic millimeter, and you'd like to do much larger and higher resolution, and both in space and time. The time is limited to about 1 second per time sample, and the resolution is typically 250 to 500 nanometers. This is work from Ahrens et al. It's beautiful real time, but we need to move beyond that. We need to get the advantages of high resolution in situ and the multi-omics that you can get in situ, but still get high temporal resolution over the entire body of the animal. So you can sample it, or you can do it comprehensively. So we've been storing data in DNA since our first proposal in 2002, which was sent to the US Patent Office. 2012, we did an in vitro chemical synthesis of a book and images, my book Regenesis, that's 2012, and then a whole series from 2016 to the present, where using CRISPR and guide RNAs, or Cas1 and 2, which is a different recording system, and then and most recently, the A to G base editor, which has at lowest toxicity and can be used at highest density. And these are sometimes called a molecular flight recorder or molecular ticker tape or DNA, flight recorder or ticker tape. This is all for temporal recording of information. One of the first ones where we got a large amount of information in vivo relevant to lineage during development and aging, all the way from fertilized egg to a severely senescent adult. It's also been adapted by Lovelace et al. I draw your attention to this Nature Chemical Biology paper in 2021, where they adapted it for physiology measurement, and you can do development and physiology in the same context. And we don't know what the upper limit to the number of physiological channels that you can record simultaneously into DNA, but it's a large number. And this can use, here we use CRISPR cutting, but we recommend, as you'll see in a moment, the A base editor. Here's an example of lineage information where we disambiguated for the first time the order of events for forebrain, hindbrain decision versus left-right decision, the latter being later. And here we calculate that it's on the order of 4 times 10 to the 12th bits of information, almost a terabyte per animal. But we'd like to move beyond a terabyte. That terabyte was established in 20 nanograms of DNA, which is 1/billionth of the mouse's body mass. So you're only dedicating 1/billionth of the animal to this amazing recording device capable of doing development, aging, and physiological measurements. But we'd like to crank that up by 6 orders of magnitude to petabyte by, instead of using 40 to 60 randomly chosen guide RNA, homing guide RNA sites, change it to as many of the points in the genome, which are either safe harbors or repetitive elements, places in the genome where the cell can continue its function without being interfered with. And we think we can get millions of such sites. And where are those millions of sites? It's in the dark matter. The parts of the genome have no known function. We will discover some function probably along the way, but I still estimate that it's we have already demonstrated endogenous retroviruses, of which there are about 60 of them in pigs, to 24,000 line elements in humans, and we're now moving on to 2.5 million combined ALU and centromere sites, and each of those sites has many subsites, so it's more than 2 million possibly. So here are the references down here in Science and Nucleic Research for the endogenous retrovirus and line element that I'll show here. So we finally have settled on deaminase of A to G as our favorite way of getting very high number of edits per cell. These are line elements, which is 24,000, but we're, as I said, going to ALU elements and centromeres. But it's critical that we did A to G rather than C to T, or worse yet, NICs or double-strand breaks, because any NIC or double-strand break, including the ones that come from mismatch repair or uracil glycosidase, are toxic at this level of editing at once. But A to G has little off-target and targeting, and we've now saturated all the line elements as far as we can tell in this particular category, 24,000 edits per cell, and this can be stochastically turned into these recording devices that we've been talking about. This is work from Corey, Oscar, Khalid, and Verena, based on a lovely work from David Liu's lab. They shared their reagents with us prior to publication, and it's been very helpful. This is now Nucleic Research gave the reference in the previous slide. And finally, I'd like to thank all the people in the world that have volunteered for our project, and the Personal Genome Project, and now all of us, and UK Biobank in England, and similar efforts that are ongoing in the UAE and China and so forth. These people that are volunteering to share their genomic, their medical records, their imaging at the molecular level, all the way up to the brain fMRI that I show in the upper left of this corner. This personal genome project has been used for the human subjects research that we did, I mentioned in this talk, plus genome in a bottle, ENCODE, and so forth. And it's now approved by ethics committees on human research in multiple countries. Period, full stop. Thank you.